Welcome to the video for chapter 6.5. So in 6.4, we talked about the different types of extremophiles and the different environments they live in. So one of the last extremophiles that we need to talk about are the ones that live in different oxygen conditions, so different oxygen environments. Okay, so let's get started. Okay, so let's do a review question. So review question number 4 says, one likely adaptation for microbes that live in a psychrophilic environment is that A, their proteins are more rigid than mesophilic bacteria, B, their membranes are more stiff than mesophilic bacteria, C, their membranes are more fluid than mesophilic bacteria, or D, they are adapted to low pressure environments. Okay, so take a second, pause the video, flip through your notes, and see if you can't find an answer. Okay, so the key to this question is knowing what sacrophilic, so what a sacrophile is. Okay, so remember, sacrophiles live at cold temperatures. Okay, so the answer to this question is C. Okay, because they live at cold temperatures, their cell membranes are going to be at risk for becoming rigid, to stopping and being hard. So what they need is proteins and things like cholesterol that are going to keep their membranes fluid. Okay, so the answer is C. Okay, so one more review question. Now this one is not one that we've talked about the answer to. This is one that I would like you to see if you can reason through based on what we know about extremophiles. Okay, and what we kind of remember from biology 201 or regular high school biology. So review question number 5 says, which of the following cellular components can be found in high concentrations in a halophilic microorganism? A sodium plus and potassium plus ion pump. B, citric acid. C, cold tolerant enzymes. Or D, a superoxide dismutase. Okay, so pause the video, go flip through your notes, and see if you can't reason through to an answer. Sorry, guys. Okay, so let's talk about this. Okay, so one of the starting off points for this question is we have to know what a halophile is. And where do halophiles live? Okay, these are the ones that live in extreme salt environments. Okay, so because it's osmolarity that we're worried about, salt content that we're worried about, we know that citric acid is not going to be one. Okay, so B is going to be out. We have not even talked about anything called superoxide dismutase, so that kind of brings D out as well. Okay, so halophilic, that means salt. Well, we don't really care about temperature-dependent enzymes because that's not going to help us in a high salt environment. So the answer is A, the sodium potassium ion pump. Okay, we talked about these back in biology. We'll talk about them a little bit in this class in a couple chapters. But yeah, we know that salts like sodium chloride, table salt, has sodium in it. There's also salts that have potassium in it. So it makes sense that if they're halophiles and they're going to try to keep themselves in a condition where they're not losing all their water, that they need to have a way to pump these different ions out so that they don't build up outside and so they can bring them inside. Okay, so 6.5 is all about different organisms living in different levels of oxygen. Okay, so our second objectives are we need to be able to differentiate between anaerobes and aerobes, and we need to be able to describe how we culture each in the lab. You need to be able to explain how both aerobes and anaerobes can cause disease, and you need to be able to discuss the basic differences between respiration and fermentation and how this impacts where an organism grows. So at this point, we're talking about respiration and fermentation in terms of energy. Which one makes less, which one makes more. Okay, in chapter 7, we'll talk about respiration and fermentation in a lot more detail. But right now, let's just think in terms of energy production. Okay, so I have a question for you. Why do you need oxygen to live? So pause the video, make a list. Maybe we'll see if you have the same answer I got. Okay, now we're going to address the elephant in the room. Ms. Harker, we need air to breathe. That's why we need oxygen to live. Well, that's a good point. But there is a lot of organisms, say bacteria, that don't breathe. They don't have lungs. So why is oxygen so important to a lot of living things if not everything breathes? Let's talk about it. Okay, so in biology, we talked about cellular respiration. We talked about the different steps in it. Okay, we have glycolysis, we have the Krebs cycle or the TCA cycle, depending on what teacher you took. And then you have that last step. Okay, so by the time we're done with the Krebs cycle, we have totally harvested all the energy that we can from glucose, and we need to then make it into ATP. Okay, so we need to store all that energy we harvested somewhere. Okay, the last step of respiration is something called oxidative phosphorylation. So the reason we need oxygen to survive is so that we have oxygen to participate in this step of respiration. So oxygen is what we call the final electron acceptor that helps us generate ATP. Now we're going to talk about this in a lot more detail in the next chapter. Okay, so just remember we need oxygen because it's our final electron acceptor. Because oxygen is our final electron acceptor in the cellular respiration pathway, it means we can make a lot more ATP than if we use something like sulfur or iron. So things that lithotrophs are going to use instead of oxygen. Okay, so because not all microbes use oxygen as their final electron acceptor, that means they're going to generate ATP in multiple different ways. Okay, and then there's going to be some microbes that live in conditions where there's no oxygen at all. So how do they make energy? Okay, in fact, these microbes that live in conditions where there's no oxygen, if they get put into an environment with oxygen, they don't have the equipment needed to deal with oxygen, so oxygen becomes toxic for them. So let's talk about this in a little bit more detail. Let's go to a case study. Okay, so this case history is called attack of the anaerobe. So we have a patient and they have extreme pain when their lower right abdomen is touched. This is never a good sign because you're going to try to figure out if what's happening there. You shouldn't have pain there normally. Okay, because they're in such pain, the ER doctors are going to recommend taking them to surgery immediately. So they start into this exploratory surgery, they open this patient up and start looking around to see what might be wrong. Well, they find that this patient's appendix is inflamed and enlarged and not in a good way. Okay, so this patient's exploratory surgery became an appendectomy. Okay, they removed the patient's appendix because of this exploratory surgery. Okay, so while they're doing this surgery, the doctors take a blood sample from the patient and then they take it to culture to figure out what bacteria is causing this infection in the appendix. Okay, so one of the things we haven't talked about, but we probably should know when we're talking about oxygen environments is the fact that our GI tract, our gastrointestinal tract, has very low oxygen levels in it. Okay, because there's very little oxygen in our GI tract, we have to grow the microbes that we pull from our GI tract, different than we would say the microbes that we've been growing in the back of the classroom. Okay, so let's talk about how we do this. Now, we have to talk about the fact that this patient had his appendix removed. Right now, our appendix really doesn't do anything. It's what we call a vestigial structure, meaning that at some point it had a function, but we have evolved and it has evolved that its function is no longer needed. Okay, so we really don't know what it does. So this is kind of the same as like vestigial legs on boa constrictors. So boa constrictors have these tiny little legs that don't really do anything, but they're still there. That's what we call a vestigial structure. So something that's not used but is still there. Kind of like we have a tailbone but no tail. Okay, so we don't know the function of our appendix besides to become inflamed and become infected and need to be removed. So let's talk about how we try to figure out what caused this infection. Okay, now I'm going to say the collection of these samples is different when we're collecting them from lab versus whether we're collecting them in a medical setting. There's going to be two different ways, and we're going to talk about that a little bit in this lecture. Okay, so the first thing we need to do is we have to be able to identify all the pathogens that might be causing this infection, because it might be more than one. So what they're going to do is they're going to take this sample and they're going to grow it in multiple conditions. So in this case, we're going to grow it with oxygen and without oxygen. Okay, so the clinical microbiologist that's in the lab who's growing these cultures found that in the bottle without oxygen, which will be this guy on the left, there's going to be no growth. Okay, well, this bottle on the right, which is the no oxygen condition, is going to have a lot of bacterial growth in it. Okay, which is going to indicate that we have an anaerobic microbe causing this disease. In fact, because of this test and a few other tests, they found that this pathogen was Bacteroides fragilis that was causing his appendix to be infected and have to be removed. Now, how we collect these samples is very important. Okay, and where we collect the samples makes it very important. Okay, so the bottle that's going to be in the air conditions, so with oxygen, we're just going to take the sample and we're going to just stick it in there. Don't worry about it. Okay, so depending on what field you're in, so for scientists, so like when I had to use these bottles when I was doing research, we would have this little like silica packet, like this little pill that we would crack and put it into the bottle, and it would drive all the oxygen out and replace it with stuff like carbon dioxide and nitrogen. Okay, so that would make it a environment with no oxygen. And that would be what would allow this Bacteroides fragilis to grow. And one of the reasons why it's a pathogen of the GI tract is because the intestines are pretty much oxygen free. So it's a great place for it to live. Now, if you're collecting samples, say in a medical environment, you're going to use something a little bit different. In fact, when we grow these cultures, so say if we want to grow it on a petri dish, this is going to be different than what we do in the back. Because in the back, we're just open to oxygen. Like we're not restricting oxygen at all. So at the end of this lecture, we'll talk about ways that we grow bacteria when we need to grow them without oxygen. Okay, so this slide and the next slide with words on it, we are going to talk about some vocabulary words. Specifically, we're going to talk about aerobes versus anaerobes. Okay, aerobes are going to be microbes that live in oxygen conditions, and anaerobes are going to be bacteria and archaea and other microbes that live in conditions with no oxygen. Okay, so we have strict aerobes. So these ones can only grow in environments with oxygen. Okay, they can't be in any low or no oxygen environments. Okay, so strict aerobes are always going to have oxygen for their final electron acceptor, which means they go through aerobic respiration, which means that they are going to probably be the most efficient at making energy. So when we have oxygen in a system, oxygen's very reactive. In fact, it causes a lot of damage. So strict aerobes are going to have special proteins, special enzymes, that their whole job is to make sure oxygen doesn't cause problems, doesn't cause damage, because oxygen can become toxic and it can become what we call an ROS. Okay, so a reactive oxygen species. So an example of a reactive oxygen species would be hydrogen peroxide. So the same thing that maybe your mom cleans your skin and knees with. Okay, so in order to deal with hydrogen peroxide in our system, we have an enzyme that's going to make it so it's not toxic and not causing damage. Okay, so strict aerobes have all the enzymes to prevent this damage from oxygen. Now, strict anaerobes, okay, we are not going to use oxygen for energy metabolism. In fact, most anaerobes are going to be going through fermentation to get energy from the food that they collect. Okay, so strict anaerobes have no enzymes to deal with our reactive oxygen species, which is going to make oxygen toxic to them. So it's going to kill them if they're in a oxygen environment. Okay, so they can only survive when there's no oxygen. Now, because strict anaerobes use fermentation, they cannot make as much energy at a time as strict aerobes who could go through cellular respiration. In fact, most strict anaerobes are going to be bacteria, so they're going to be very small. Fermentation barely makes enough energy for them to function. Okay, and then you're going to get other conditions like when you go through fermentation, you're going to make either alcohol or lactic acid, which can then also change the environment around you if you're going through fermentation. So these are some important differences to know. Now, this is a very, very good diagram to know. Okay, so we have this test tube with different oxygen growth regions. Okay, so for aerobes, we're only going to find them in this very top section where there's a high amount of oxygen. Strict anaerobes, we are going to find here at the bottom of the test tube where there's very little or no oxygen. Okay, so what grows in between here? So we call this region the facultative region, or where we're going to find facultative or aerotolerant anaerobes. Okay, we also have a class that are called microaerophiles. So let's talk about the difference between the ones that grow between the high and the no oxygen conditions. Okay, I would definitely know these definitions along with strict aerobe and strict anaerobe. Okay, be able to tell where they can live on the test tube. Okay, so the thing with facultative anaerobes, microaerophiles, and aerotolerant anaerobes is that they can live in both conditions. They can have a little bit of oxygen, they can have some no oxygen conditions. They're comfortable in both. Okay, so facultative anaerobes, these are the most versatile guys. They can do fermentation or respiration. So they can actually do both aerobic and anaerobic respiration, and we'll talk more about this when we talk about respiration in the next chapter, what the difference is. Okay, now, in terms of energy production, fermentation makes the least, anaerobic makes the next most, and then aerobic is the most efficient and the best way to make energy. Okay, so for facultative anaerobes, where they can do any of these three processes, they can survive with or without oxygen. They're always going to have a means to make energy. Okay, so microaerophiles, okay, these are going to be mostly aerobic, but they are not as well equipped as strict aerobes. Okay, meaning they don't have all the enzymes needed to combat those reactive oxygen species. So some reactive oxygen species like hydrogen peroxide can be toxic to them. Okay, so they prefer environments where there's a low oxygen concentration, so it's not as toxic to them, or anaerobic conditions where there's no oxygen. Okay, so microaerophiles are going to be closer to the top of the tube than the bottom. Now, aerotolerant anaerobes, these are going to prefer to be anaerobic. They can live in low oxygen conditions. They're less susceptible to reactive oxygen species than microaerophiles are, but they usually lack catalase. So catalase is the enzyme that protects you from damage from hydrogen peroxide specifically. So if you ask people in the first aid community what's the best way to clean out a wound, say a scrape or something, they're actually going to argue about whether you should use isopropyl alcohol or use hydrogen peroxide. Now, I know, like in my family, I have family members that think hydrogen peroxide is better because it foams, right? If it's really dirty, it's gonna foam a lot. That's not because it's actually cleaning your wound. It's because there's catalase reacting with it. So the catalase is neutralizing the threat from the hydrogen peroxide, and you might get the side benefit of it cleaning out your wound, but it's not a great way to do it because you're actually causing just as much damage. Okay, so with aerotolerant anaerobes, they can survive in oxygen conditions, but they prefer to be in no oxygen conditions. Okay, and for the most part, they use fermentation because they prefer to be in an anaerobic environment, so they don't have oxygen as that final electron acceptor. Okay, so this is a fun table from our book with some very specific examples of aerobic and anaerobic microbes. Okay, so some example of aerobic microbes that we've talked about, Mycobacterium leprae, so the one that causes leprosy, it's aerobic. The Neisseria species, so these mostly cause meningitis and gonorrhea. These are going to be aerobic. Our good old rhizobium that is in a symbiotic relationship with legume plants like pea plants, is going to be aerobic. Okay, so anaerobic ones are going to be things like the Clostridium species, so like Clostridium botulinum and Clostridium tetani, so the ones that cause both botulism and tetanus. Okay, so these are going to be found in soil. They don't like to be in high oxygen conditions. They like to be with no oxygen. Okay, we have our facultative anaerobes, so they can live in either condition. They don't really have a preference, so like a lot of the Staphylococcus species, so like our Staphylococcus epidermidis that's found on our skin, the different Staphylococcus aureus, which causes staph infections, also facultative, can be inside or outside the body to cause those infections. Bacillus anthracis, Vibrio cholera, E. coli, these are all facultative anaerobes. So one of my favorites is actually a yeast. It's Saccharomyces cerevisiae. This is what you put in your bread to make it all light and fluffy and create those air pockets. So Saccharomyces cerevisiae is going to go through fermentation, and when it goes through fermentation, it's going to create carbon dioxide, which is going to create those nice light air pockets in your bread. And it's going to also produce lactic acid, but that's going to cook off while you're making your bread. Now, some common microaerophiles are going to be things like Helicobacter pylori. Okay, so that acidophile that causes ulcers. Lactobacillus. This is going to be the bacteria that makes your yogurt. So it's going to do fermentation to make that lactic acid, which is what makes your yogurt taste so good. Okay, and then we have Treponema palladium, the organism that causes the STD syphilis. It's also microaerophilic. Okay, so with aerobes and anaerobes, we have to be very careful about sample collection. In fact, it really does matter how you collect these samples. Okay, so there's different ways to collect samples. So we talked about the little bottles, the one with oxygen and one with the no oxygen conditions. That is mostly scientific sample collection. Let's talk about what happens in the medical field. Okay, because one of the things in the medical field what we do is we're going to use these samples to do a bunch of diagnostic tests to figure out what's causing disease in a patient. Okay, so if you go into the medical field, you are very likely going to be the one collecting the sample and helping diagnose these patients. So for different types of wounds, we collect different types of samples. So if it's a surface wound, so something that's open to the air normally, those are going to be aerobes, we're just going to swab it like normal. But if it's an internal wound or it goes pretty deep, where it could possibly have low oxygen or no oxygen conditions, we are going to treat that swab differently. Okay, so in the medical field, they're going to have a wound swab with this test tube. This test tube is going to have like this gel-like media in it, this transport medium that we can stick the swab in after we swab an internal wound that's going to put it into an anaerobic environment, so that gel's not going to have any oxygen in it. Okay, so to collect the sample, we're going to swab the wound. So we're going to use this zigzag motion and go back and forth until we've gone through the whole wound, and then we're going to stick that swab back into this container and make sure that the sample collection end is firmly covered by all the media at the bottom that's going to keep it in an anaerobic environment. Okay, so with something that's on the surface, we're going to still swab it in the same way. We're going to go back and forth, but we're just going to stick it into a tube where it's going to stay sterile and not contaminated, and it doesn't have the transport media in it. Now, so this is something from Dr. Burroughs. Dr. Burroughs found these, what is it called? So he found some things from a nursing class. This nursing class is Nursing 113, and it's literally talking about obtaining a wound drainage specimen, so collecting samples. So this would be something that if you're deciding to go into nursing that you could possibly do. Now, I'm just going to point out, we're not going to read through everything, but it's going to reiterate what we've been learning about in this chapter with sample collection, is that we're going to have to use different techniques for collecting samples in aerobic versus anaerobic conditions. Okay, so aerobic organisms grow in the superficial wounds exposed to air. Anaerobic organisms grow deep within body cavities where oxygen is not normally present. Okay, so this course is also going to have a bunch of things. So this goes through how to collect the wound, how to swab it, the different techniques, okay, the different things. So this is just a bunch of examples of what you could see in nursing school. That's based on what we're learning in micro right now. My favorite here is how to collect these. If you take samples from different areas, you got to place them in different containers. You can't put them all in one because you might have to have different types of culture. So if you take a surface one and an internal one, you need to make sure that they're in both the aerobic and anaerobic conditions. And what I love is good old hand hygiene. Okay, we need to make sure that we're washing our hands using gloves, not just in the medical field, but in the lab too. Okay, so this is going to be a lecture that actually has a lot of real world connotations, a lot of real world applications for those of you who want to go into the medical field. Okay, so the last thing we're going to talk about is how we grow these little guys in the lab. We can't do this in our classroom. We don't have the material. This is actually quite spending material. I have used one of these to grow anaerobic bacteria, but it's been a long time. The easiest and cheapest way to grow anaerobic bacteria in the lab is to have an anaerobe jar. Okay, so this is for petri dishes and for test tubes. So we're going to have our jar. Okay, so it's going to have a seal. Okay, and it's going to have this valve. So what we're going to do is we're going to do a lot like that previous bottle. We're going to have something called a gas pack that we're going to put in, and it's going to do a chemical reaction where it generates hydrogen gas and carbon dioxide gas, and it's going to push the air out. So when you seal it finally, there's going to be no air inside the anaerobe jar. Okay, so it's going to remove the oxygen. Okay, so then this is the cheap guy. The really fun guy, this is what I've used actually, is an anaerobic chambers or an anaerobic glove box is what we used to call it when I was in school. So this is going to be a lot like the fume hood we have in our classroom, except it's all enclosed. It's going to have an airlock where you can put the sample in and then evacuate all the air using a vacuum pump. And then you're going to use a mixture of nitrogen and carbon dioxide gases, and you're going to pump them in to the chamber to make sure that there is no oxygen left in the chamber so you can do your work and that your research won't be affected by oxygen where it can potentially kill the species of bacteria you're working with. Okay, so we do all of our work with these gloves. This is also an incubator, so it could be turned on like our incubator in the back of the classroom. This is harder than it looks because you put your sample into the airlock, you've evacuated it, then you gotta put your arms into the gloves, then you have to open the airlock, and you gotta get your sample out, and then you have to do all the things that you'd normally do with the sample out on the countertops inside the box. It's fun and annoying and cool and exhausting all at the same time. But unfortunately, not even CEI has these things for us to work with. They are really specialized equipment. Okay, so that is chapter 6.5. We talked about what lives in what oxygen conditions. Definitely know all of the different types of aerobes and anaerobes and be able to identify where they can grow. Specifically, know that test tube example. Okay, so we will see you for the video for 6.6. Where is this?